Thank you, everyone. We're going to start this session. We have an esteemed panel here with us today. Everybody come on and get seated. So this panel is regulatory pathway and applications in drug development for rare disease and case studies. So the purpose for this panel I alluded to yesterday. There's been a lot of recent approvals that, they disseminate into the broader community. There's, misconceptions sometimes about the totality of evidence that get discussed in the broader community. So what better way to address this was to assemble a group of people that could speak to different elements of, recent approvals. Obviously, there are some individuals that can't speak directly to their product, but they can talk through the steps on this panel. But what we wanted to do is address those so that people would get an opportunity to see, and hear examples of the dialogues and conversations, but also the levels of evidence that are needed to be done. So I'm going to start this panel with continuing the conversation we had regarding Reata, and Shannon, if you want to interject anything additional after you hear comments from the agency. We have Emily here, who actually did the review and can provide some background experience, and we'll come back and we'll interview people. And after this, I just want to start that for continuation, and we'll go down the row.
Sure, thanks. So for those that don't know me, I'm Emily Freilich. I'm the director of the Division of Neurology 1 at Cedar. And this is a good continuation since we just heard a lot about omaveloxolone and Reata. And I think Dr. Baracchio also mentioned yesterday some of the considerations that went into determination of substantial evidence of effectiveness for that application. As you heard, we have to determine substantial evidence, and typically it can come from two studies, a single study, or a single study plus confirmatory evidence. And as we also heard, Reata, omaveloxolone did have a positive, adequate, and well-controlled study that won on its primary pre-specified endpoint. But it had some limitations as well. It was a small study. It did not have positive findings on the secondary endpoints. And so it wasn't persuasive enough to stand alone as a single study. And so we really were considering what could serve as confirmatory evidence to support what appeared to be a positive treatment effect in the single pivotal study. And they had presented also some mechanistic data that showed that the drug seemed to be doing what it was purported to do. So some biologic plausibility that the drug was having its effect on NRF2 activation. And the natural history study, which we heard a lot about in the last session, but the benefits of that study, or the positives that allowed us to really rely on it, were what we heard a lot about was the natural history database, right? This FACOMS, where we had individual patient-level data and from a large number of patients that had systematically collected the same mFARS that was the endpoint in the primary, in the open label extension for the pivotal study. And so the other thing that was also compelling was that a lot of the centers were the same, and so we felt confident that the data was collected in a similar fashion. I think there was overlap on a lot of the centers between the natural history study and the open label extension study. And so looking at that data and seeing that you had several years of progression in the natural history data and could compare it to the 3 years of open label extension data on the same endpoint on the mFARS, and we're able to do the propensity matching because they had collected demographic data for baseline characteristics to be able to match on the important prognostic variables.
Slowed some people from getting here. So for the first session, I want to have them introduce themselves. We're going to have an FDA Fireside chat looking at the impact of rare diseases and FDA's perspective on them. And they're going to convey some of the programs that the FDA is undertaking, and this will be followed by a Q&A. Really exciting activities going on in agency, and we want to make sure to introduce those with the team. Who might be introducing slides first?
I guess I should start, yeah. we have a clicker? Okay. Perfect, thank you. All right, good morning. I'm Jacqueline Corrigan-Karai. I am the Principal Deputy Center Director in Cedar. It's really a pleasure to be here. I'm not sure if I'm getting an echo because I have 2 mics on. Am I okay? All right, good. So what I'm going to do is just talk a little bit about the landscape for rare diseases in Cedar, and then I'm going to hand it over to my colleagues to talk about some more specifics. So here we all are, and I'll have each of them introduce themselves as they come up. So these are just some numbers for you, and it really shows you the Cedar orphan drug approvals of new molecular entities by calendar year. The last year in 23 is only through September 6th, so a little bit lower. But you can see from, 44% to 52, but on, about half of all our NME approvals are for rare diseases, which is really encouraging. I know there's more work to be done, but certainly. And importantly, many of them are able to take advantage of what we call our expedited programs for development, including priority approval or priority review, actually, which really means that when it comes into the FDA and we have preliminary, we can say that there's going to be a significant improvement, that we can rapidly accelerate a little bit the review of that package. Fast track is something that you can get based on preclinical data as well as clinical, and then breakthrough therapy is when you have preliminary clinical data that shows that there may be a substantial benefit over what's available. Now, as you can see, about half of them get either one of those. Accelerated approval, of course, is a little bit lower, and that's really because you need a biomarker. You need a biomarker that we say is reasonably likely, so that's one of the limiting factors in that one. So one of the things to remember, we have a number of initiatives that are regulatory science initiatives, and they span, they're not always rare disease specific, but they can be incredibly important for rare disease development. For example, complex innovative trial designs, when you're thinking about designs for smaller trials. The critical path innovation meetings, if you're thinking of something new, you want to get some early, this is not a pre-IND, this is really to discuss your ideas with us. Model-informed drug development, and then, of course, patient-focused drug development. I think many of you may be aware of. Many of those sessions have been around rare diseases, and now I believe all 4 guidances are out so that you can really think about how to integrate the patient perspective in drug development. We support RDCA-DAP, very important, getting that data together, getting it in a structured manner that we can really learn from. And I'm going to talk a little bit about the last 2, which is real-world evidence and digital health technologies. We often think of real-world evidence as big data, but I think it's really what real-world evidence is trying to use every piece of data that we have from clinical care and see if we can apply those learnings into the regulatory. And then, of course, digital health technologies. So in the real-world evidence program, we've put out a number of guidances, and I think I'll highlight several that are probably quite relevant. One is our externally controlled trials for drug and biological products put out just this year. Now, I'm not saying that every rare disease development program should be an externally controlled trial. We often need randomization, but just if you are going to be taking that approach, this really gives you some important guidance on what to consider in terms of what kind of data, the comparability issues you have to think about, analysis considerations, and then considerations to support regulatory review. The other guidance I just want to highlight, which was done a couple of years ago, and we're finalizing it right now, is the registry. So, again, registry is quite important. And so how should you think about them and using them for regulatory decision-making? How should you design them, actually, is in this guidance when you're thinking about that. And then how do you do linkage, and what do you have to think about when you're trying to link a registry to outside data, EHRs, or digital health technologies, and then how we might review it and kind of thinking about what data needs to be submitted to us. So I think those are important guidances to be aware of. The other thing is there's been a lot of attention on digital health technologies. We published a guidance on this, I think it was almost 2 years ago, that we had a number of comments, hundreds of comments. So there's just great interest in the use of digital health technologies. And then under PDUFA 7, we have a commitment, and we published the framework for the use of digital health technologies in drug and biological development just this year. And so this is really thinking about how do we change trials? How do we think about collecting endpoints, make trials more accessible or different endpoints, really understand the patient's journey outside of perhaps their visits. And that really gets into thinking about, can we do trials where at least some or even all of the trial-related activities take place? Again, that'll depend upon where you are in your development program, what's the intervention. But thinking about how do we make trials more accessible. And towards that end, we just published several months ago the guidance on decentralized clinical trials for drugs. And so really thinking, is it video and telemedicine visits? Is it direct distribution of the product? How do we make electronic informed consent more easy, home visits, and of course, integrating digital health technologies. So all of these are, innovations that, can be used in the rare disease space, especially when you're thinking about diseases where, geographic recruitment may be quite wide. So this is just an overview of some of the regulatory science initiatives that are not rare disease specific, but certainly can be leveraged by the rare disease community. And now I'm going to turn it over to Carrie Jo, who's going to speak a little bit more about our newest program that is quite specific for rare disease.
Thank you so much. Hopefully everyone can hear me without an echo. Sounds like it. Thank you. Sorry, I'm just trying to advance this slide.
The green one, the big green one.
Did pick green. Oh, big green. Now we'll go back one. There we go. So back in May of 22, CDER really wanted to consider how can we harness all of our collective experience and collective knowledge to really try and accelerate rare disease drug development from the regulatory perspective. And to that end, we decided to launch the Accelerating Rare Disease Cures Program, or the ARC program. The vision of this program is that we could speed and increase the development of effective and safe treatment options to address the unmet needs of patients with rare diseases. And the mission for this program was really to drive the scientific and regulatory initiatives and innovation and engagement to accelerate the availability of treatments for patients with rare diseases. As you saw from the previous presentation, this represents a substantial degree of the work that we do at the FDA when it comes to new molecular entities and programs. So what does the ARC program look like? Well, it's a program that has come together and is governed by senior leadership from the areas of CDER that are most directly involved with regulatory drug review. So that would be the Office of New Drug that houses your clinicians and your pharm tox representatives. That has the Office of Center Director, where you have senior leadership, such as those who are speaking with me today, as well as the Patient-Focused Drug Development Program. And then you have the Office of Translational Science, which houses our statisticians, our clinical pharmacologists. And bringing all of these senior leaders together to govern the ARC program and look as an umbrella program over those initiatives that are specific to rare diseases, as well as those that can inform rare disease drug development, was critically important. The program is managed by the Rare Diseases team, and I realize I did forget to introduce myself. I'm Dr. Carrie Jolie. I'm the Associate Director for Rare Diseases in the Office of New Drugs, and I lead the Rare Diseases team. Our team is PDUFA mandated to carry forth and connect the rare disease programs for CDER. Now, when you look at CDER's ARC program, you have the ARC governance board and you have the rare diseases team which manages it. But it doesn't stop there. It is an umbrella program for the center. And therefore, we work very closely with our colleagues across other offices, such as the Office of Medical Policy, which has a lot to do with benefit risk and real-world data and real-world data and real-world evidence and digital health technologies. You have the Office of Surveillance and Epidemiology, which additionally is informative to our use of real-world data and real-world evidence, as well as post-marketing safety. You have the Office of Strategic Programs, which is where a lot of the benefit risk work is centered. And then you have other offices, CDER offices and divisions that may contribute to all aspects of rare disease drug development for subject matter expertise as necessary. And then we also work closely with other FDA offices and programs. So that includes our good partners at the Center for Biologics, evaluation and research. We have many similar indications and conditions to manage, and we work with them very closely. We have a Center for Devices and Radiological Health. We work with the Oncology Center of Excellence. We work with the Office of Commissioner, specifically the Office of Orphan Product Development very closely, as well as the Office of Pediatric Therapeutics and the Patient Affairs staff. So ARC is really an umbrella that pulls together all of these groups in order to really drive our focus to develop methods and innovation to aid rare disease drug development. And I have our website on there, which I encourage you to go to. So what are some of the things we really focused on in our first year of development? This was really a focus on engagement. It is critically important that we share our regulatory thinking about the challenges and conditions that affect rare disease drug development. And to that end, there were 4 workshops held under the ARC umbrella the first year. The first one was in conjunction with our partners at the NIH in regulatory fitness for rare disease clinical trials. And that was a conference that spanned many, many aspects of drug development and lives on in perpetuity at our website, as well as the FDA Duke Margolis virtual public workshop, which we had on translational science development, specifically on surrogate endpoints, biomarkers, and more, which are critical to rare disease drug development. Many of these conditions are slowly progressive, and to try and measure a clinical endpoint during the duration of a trial can be extremely challenging. So this conference really focused on how to develop those endpoints, what are the challenges, and what are some potential solutions, as well as examples. We additionally held a CIRSE in 2023 of May, and this was one that was very, very timely. It had to do with addressing challenges in design and analysis of rare disease clinical trials, specifically focused on the use of real-world data to inform the design of rare disease trials, as well as challenges in analysis with these small populations, so statistical challenges. And finally, there was another CIRSE held with the University of Maryland on quantitative systems pharmacology and some of the more advanced modeling that can be done. Additionally, it's incredibly important that we engage with our patients. So we engaged in 23 rare disease patient listening sessions that first year, held between patient affairs or the professional affairs and stakeholder engagement, and 19 patient-focused drug development meetings. So this is just a way in which we are trying to engage more and listen to our patient stakeholders about what is important to them about their conditions that can help to inform rare disease drug development. And then finally, I'm just going to end with a plug for our quarterly newsletter. We are trying to get the word out as much as possible on rare disease happenings that the FDA is participating in. So I would ask that everyone go through and sign up for that, and you'll find out about things. All of the workshops that I mentioned are on our website. They live on in perpetuity so that people can go and educate themselves on different aspects of rare disease drug development. And now I'll turn it over to Dr. Baracchio.
Green. Okay. Hi, I'm Dr. Teresa Baracchio, and I'm the acting director of the Office of Neuroscience in Cedar. And I'd like to take some time to discuss 2 rare disease actions that we've had this year that have been impacted by some of the initiatives described by Dr. Corrigan-Kurai and Dr. Lee right before me. The first was the approval of omaveloxolone, which is also known as Skyclarys, for the treatment of Friedreich's ataxia. And this is the first treatment that's approved for this disease. Friedreich's ataxia is a neurodegenerative disease that begins in childhood and has a very heterogeneous course and causes impairment in motor function. So the approval was based on substantial evidence of effectiveness based on a single adequate and well-controlled study with confirmatory evidence. The sponsor conducted a adequate and well-controlled 48-week randomized controlled trial that had a clinician-reported outcome that was positive, although there was not strong support from secondary endpoints. Although the study was positive, it was not strong enough to stand on what our usual standard would be for a single trial approval alone. So we do have a pathway that's available to us where we can utilize confirmatory evidence that would substantiate the findings seen on the adequate and well-controlled trial. So we worked with the sponsor to identify confirmatory evidence that could support the primary evidence from the controlled trial. When we work on these programs, we do take into consideration the rarity of the disease, the unmet need, and the challenges in the ability to conduct a second trial. And we felt that it would be quite challenging for the sponsor to conduct a second adequate and well-controlled trial in this disease. So we were willing to work with them to try to figure out a path forward. In looking at the confirmatory evidence that the sponsor proposed, they had proposed to use mechanistic data to support the drug's mechanism of action that was relevant to the disease pathophysiology. They conducted a delayed start analysis using the open label extension data compared to the controlled period of the study. And they used, took their open label extension data and they compared it to a natural history study, which was the FA-COMS. No single element of these was served as a piece of confirmatory evidence alone. We looked at this as a total package, and I think that's one thing when you look at our guidances. We have discussion of confirmatory evidence in the 2019 effectiveness guidance. And when you look at that, you might be led to believe that you need to have a single piece of confirmatory evidence, but really we can look at multiple components for confirmatory evidence, and overall we put a package together that we think is compelling to help substantiate the effects seen in the primary study. I will say that much focus has been put on the open label extension compared to natural history comparison, and I've seen a lot of comments about that in the press, but I will just say, although we did utilize that data, and I don't want to downplay that, but that was not the primary evidence of effectiveness. The primary evidence of effectiveness was still a positive, adequate, and well-controlled trial, but we were able to leverage the open label extension data, the natural history data, to help support that approval. And I'm not going to go into details of that now because I believe I will get an opportunity to talk about this more tomorrow at another meeting or another session. So I'll save the details of that for that meeting. The second approval that I want to discuss is tofersen, which was an ASO that targets SOD1 protein. And it's also known, the brand name is Qalsody, I guess. And it's approved for ALS due to SOD1 genetic mutation. So this program received accelerated approval based on reduction of plasma NFL neurofilament light protein, which was found to be reasonably likely to predict clinical benefit. A phase 3 study was conducted that was an adequate and well-controlled study, and it was a fairly large study for this population. It enrolled 108 patients, and overall it's estimated that there's just about 300 patients with this genetic mutation in the US. So 108 patients is a large study for this population. And they treated patients for 24 weeks. The study at the 24-week or 28-week was the analysis point. At the 28-week point, the study was negative on clinical endpoints in the controlled study, but robust reductions in NFL protein and SOD1 protein were seen at that point. And when you looked at the open label extension data, there were notable trends for clinical benefit if you looked out longer to a 48-week period. So it was enough to suggest that this 28-week trial was not a negative study per se, but rather a failed study. And there were reasons to think, to see why the study failed. One being that it was a small study and likely underpowered for the clinical endpoint, and also that the study duration was too short. So there were reasonable rationales for why this study would have been considered a failed study and why there may still be evidence of effectiveness for the drug. And also it was a targeted therapy that was known to impact the underlying pathophysiology of the disease. So based on this, we felt that the data was not sufficient to support a traditional approval, but that with additional analyses of NFL, that the reduction in NFL in the context of a targeted mechanism and SOD1 reduction, that NFL could be considered reasonably likely to predict clinical benefit. And so I just want to emphasize again that it's important to note the context of this the use of this NFL, which was that this is a population with an identified genetic target, that the drug targeted that genetic mutation, it showed target engagement with SOD1 reduction, and that the NFL reduction was felt to be a likely downstream effect of reduced neurodegeneration related to the disease. So that context is very important because, again, one of the things that shows up in the press is we've used NFL as a surrogate endpoint, and are the doors wide open for NFL? I think NFL is promising, but it does really need to be considered in the context of the disease and the development program and the therapy. So we're not ready to open the doors completely for NFL, but I would just say it's quite promising, and we think it should be considered as an exploratory endpoint in many of our neurodegenerative disease programs. And one other caveat before I step away was that the other thing that I've seen a lot of press about with this approval is that we approved a drug with a negative study. And so I do want to emphasize that it's not typical that we approve drugs with negative studies, but if there is a strong rationale for why a study failed and you have a strong scientifically based hypothesis to support the analyses that are done in the case of NFL and SOD1 reduction, that we can still look at the data. This is not a fishing expedition or cherry picking of data. If you have a really strong scientific rationale behind it, that should be pre-specified before you start doing your post hoc analyses. But I did want to just comment on that, as I know we've seen a lot of comments on that in the press. So I think with that, I'll stop and turn this over to Dr. Celia Witten from CBER.
Thank you. Can you hear me? Yes, okay, good. Yes, thank you, and thanks to the organizers for inviting me to speak. I'm going to talk about rare diseases at CBER and a couple of initiatives in CBER for rare disease development. But first, because I think this audience may be more or less, some of you may be less familiar with what products are regulated by CBER, the Center for Biologics at FDA, I thought I'd just start with this slide. Allergenics, blood products, tissues and cellular products, gene therapies, vaccines, xenotransplantation products, certain devices, and then some other miscellaneous categories. And I think to this audience in particular, gene therapies is important, although some of these others are also for rare diseases, especially, for example, xenotransplantation products. So just keep that in mind too, but most of this topic will focus on gene therapies. So we do have a rare disease program at CBER, formally with the lead for the rare disease program. Some of you may know Julie Valancourt. I don't know if she's here today, but obviously it's smaller than CDER's because we're relatively smaller. And we do a lot of active collaboration with outside stakeholders and collaboration with our colleagues at CDER on projects and on policy development. But we have our own policies specifically for some of our products that we're developing too, and I'm going to talk about them a little bit later in this talk. We also have a patient engagement program with the lead, Karen Jackler, who's actually attending this meeting with me today. But again, it's a smaller program than what is at CDER. So this is the office that regulates cell and gene therapy and some other products too, is actually been reorganized and is now called the Office of Therapeutic Products. But this slide is from when it was called the Office of Tissues and Advanced Therapies. And I'm just showing this slide to give you an idea of the explosion in interest in cell and gene therapies, which mostly drives this over the last couple of years. And you can see for yourselves, I don't really need to read the numbers. The blue bar represents the investigational studies, and the orange part of the bar represents continued access programs or access programs. So I mentioned that we have some specific policy development that we do or need to be working on in the cell and gene therapy area. And PDUFA 7 gave these specific obligations. So in the area of patient-focused drug development, we have completed this one. We had a public meeting November 15th on patient perspectives on gene therapy products and issued a summary report on that. Novel approaches to development of cell and gene therapy. This requires draft guidance on evaluation of efficacy in small populations, and also separately, a question and answer draft guidance on FAQs from sponsors. I think these two are really going to be of a lot of interest to the public. The first one is just exactly what it sounds like, and as you would expect, there are certain differences in development for different kinds of technologies, and certainly for gene therapy, it's appropriate to have a draft guidance that focuses specifically on cell and gene therapy in small patient populations. So we're actively working on, at least at this time, thinking about the scope of that guidance. Question and answer draft guidance on FAQs from sponsors. So many of our products are quite novel, but after a product is in scientific development for a while, and there are other products like it, we may have a lot of questions from sponsors which we have answered enough times that we can give, it'd be useful to the public for us to give that answer to the public. And so we have a project where we're looking at all the kinds of questions and answers we've been asked over some period of time to try to develop this guidance. So that is an active development. And then this next requirement has been completed. We held a public meeting on methods and approaches for capturing post-approval safety and efficacy data on cell and gene therapy products. The other obligations are to update the guidance on expedited programs for the development of regenerative medicine therapies, including, for example, the RMAT designation, and also, I think, really importantly, leveraging knowledge, a meeting and draft guidance on leveraging prior knowledge and public knowledge across therapeutic contexts. This is something that certainly comes up. I know it comes up across FDA, but it certainly comes up specifically in the cell and gene therapy area, meaning, what have we learned that will help us develop future products? Obviously, for an area like cell and gene therapy, that's critical, both for the clinical development part of it and also for the CMC and preclinical development. So now I'm going to move on and talk about one regulatory pilot that we are undertaking at CBER that will be announced later in the year, or at least implemented, I hope, later in the year, originally called the Communications Pilot, or Operation Warp Speed for Rare Diseases, but I think it may have some other name by the time it gets announced. So Warp Speed was the term used for development of the vaccines, which were really developed for COVID in a very quickly, given how complicated an enterprise it is. And so the question is, can we similarly speed up the development of product for rare diseases by taking a look at how we do things? And one of them is communicating with sponsors. So this gives you the idea of the history and the purpose. The products eligible will be products for life-threatening rare genetic diseases showing promising efficacy early in development. And the benefits of the sponsors will be an initial meeting followed by ongoing informal interactions via email or live meetings on an as-needed basis. So you'll hear more about this later in the fall. The last two things I want to mention is we have a very active stakeholder engagement program and a very active program for regenerative medicine education for more aimed at patients and families. Although both of these programs are extremely widely attended, and we have, for the stakeholder engagement, the virtual town halls, which are really aimed at developers, we have a lot of patients attend, and vice versa for the regenerative medicine education series. So these are launched in September 2022, and they engage with product development stakeholders in a question and answer format to provide regulatory information to advance drug development. As I said, we've had really overwhelming attendance. And for some of them, and we get questions during the event, some of them are pre-submitted. To one of them that we had in April of this year, there was such an overwhelming interest and such great questions that were asked that we had a meeting in April on gene therapy, CMC, and controls. We basically had a second one on CMC cell therapy, manufacturing controls, and then more recently one on preclinical development. So I think that's, I'm not going through the slides that list what all the different events are that we had, but if CPATH posts these, I have links included at the end of the slides. They're easy to find even if the slides aren't posted. So then this is the second educational series that I mentioned, Regen Med Ed, Patient Engagement in Regenerative Medicine. And this is an educational workshop and webinar series to engage patients, caregivers, and advocates to discuss regenerative medicine therapies, explore opportunities for stakeholders to help advance product development. There's a high attendance rate. These are virtual meetings, so an attendance rate of 63% is high. And we haven't really done, I'd say that the attendance is high both from word of mouth, stakeholder outreach, and listservs, but it is very much attended. Some of the topics I think are quite interesting. One, it started out as a basic FDA CBER workshop for patient advocates, and then there was a seminar on Regenerative Medicine 101 just to explain what these various therapies were. A webinar on the critical role of patients in advancing gene therapy treatments for rare diseases. And there's some other topics also I'm not going to list, but one that I found really interesting was the April workshop was on clinical trials, the patient experience, where we recruited patients or family members to describe their experiences with participating in clinical trials. I think that's important, honestly, not just for patients, but for developers too. I mean, I'm sure that they do have a good idea of what it is for patients, but it's I think, always appropriate to think about what it means to be in a clinical trial from a patient point of view. So that is the end of my talk, and I'll just stop here and take any questions along with any others. Thank you.
Thank you. Well, we'd open up the audience for questions at first, and then we've got a few, if there are people who want to come to the mic. One question I think that I'd like to ask the panel that came up on another discussion point is that there's rare disease obviously is an international landscape. What type of collaborations or things that are you doing globally to connect with other jurisdictions around rare disease drug development? Would anyone want to make comments to that?
I think I'll turn it over to Carrie Jo, who does a lot with their clusters. Go ahead.
I can start with that. So rare disease really involves a global community, and we are very well aware of that. And we have extremely close coordination with our colleagues through a number of mechanisms, one of which is the International Rare Disease Cluster. And that is a cluster that involves the FDA, the EMA, as well as Health Canada. And we meet regularly to discuss development programs, challenges in rare diseases, as well as exchange information about initiatives and regulatory innovation that is going on within our space. And so that is one of the ways in which we do that.
Sounds good answer.
Yeah, will also add that specifically because cell and gene therapy are sometimes have some additional challenges. There's specifically also a cell and gene therapy cluster. And we have a number of other kinds of international educational efforts or capacity building for regulation, which I think are perhaps probably shared with CDER.
Okay, thank you. The other, I guess, one of the other things that I'd like to add to this is that in addition to this room, there are a lot of impacted individuals that are on this call that can directly ask questions. So one of the things I think that you all highlighted was the FDA's interest and support of engaging the patient community. And I wanted to know if, perhaps you could walk through what that experience or give some examples more concretely than what we're giving on the slide so that people will know what those meetings look like, what types of things do you talk about, what are the venues, so that as they're thinking through their own situation, they may be able to extrapolate, what that dialogue might look like rather than just starting from scratch.
So I don't know if you want to, so there's both patient-focused drug development meetings and also what I
Patient
listening sessions. So slightly different. I think some of the patient listening sessions are really driven by the patients to tell us a lot about their experience and with the disease and what's important to them, which is incredibly, I think, reviewers get a lot out of hearing directly and really understanding. I don't know if you want to talk a little bit about patient-focused drug development, how that might be a little bit different.
I can, and I think what's critically important, a lot of these rare conditions, they can be really heterogeneous, right, in terms of the clinical manifestations, in terms of the experience of the patients. And so what gets discussed at, per se, a patient-focused drug development meeting is where the FDA review staff and communication staff really comes and is able to engage with patients representing a specific condition about which aspects of their condition are the most impactful to them. What is their unmet need? How do they feel about various aspects of how to best participate in clinical trials? And the FDA staff really listens to that, and it makes a difference when we go back and review a trial or help engage with drug development companies throughout the process of developing a pivotal trial, that we've heard from patient groups and can consider what is really going to be the most impactful from their perspective.
I can add to this. So with the two different types of meetings, the patient-focused drug development and the patient listening session, emphasize that both of these are patient-run events. So the patients do the speaking, they drive the agenda. FDA attends, but is largely in listening mode. It's not there for a back and forth on regulatory issues. It's really for us to hear and absorb the patient experience. The patient-focused drug development meetings and the patient listening sessions are quite complementary. They are different, but they can complement each other. The patient-focused drug development meetings are usually organized events at a site. Depending on where it is, FDA may or may not be able to attend in person, but we at least try to attend virtually if it's not in a local area. But there's often panels of patients, preset questions, Q&As of surveys and things like that. So it's a little more formal in its structure, and it's usually a day-long event. And we do listen to those, and then a patient voice summary is written up out of that. But a patient listening session is something that's directed to the agency directly and happens on agency time. It's usually, lately since the pandemic, it's been virtual, although prior to the pandemic we had done a few inpatient. And it's usually a smaller group of patients who are expressly describing their lived experience with the disease. And so there's often 5 or 6 participants, and they tell their stories. And then at the end, the FDA may ask some questions just about specific symptoms or things like that. But again, it's not a forum for regulatory advice. It's really a forum for us to listen and hear. But I find them invaluable. I know our staff does as well.
No, thank you. I think the other question, I think, we've discussed quite a bit, and I think it was alluded with the accelerated approval pathway. There's a lot of supplementary tools around supportive evidence, and I don't want to go in great detail today, but if there are any pearls that you all want to convey around this, because there's been a lot of, I think, a little bit misconceptions about biomarkers. Michelle's favorite term, digital biomarkers, and then use of real-world data, supportive evidence. And are there any initial pearls that you might want to share with the audience around how you consider those in the review process?
Yeah, I think my general advice is to think about this very early in the product development pathway, that surrogate endpoints are sort of a holy grail, but can be hard to develop and validate. But you can use biomarkers throughout development for various uses, even not necessarily in support of regulatory use, but for drug development in the space of proof of concept and early discovery. But later they can be used in drug development to help define the population, to enrich the population, to measure response, and then possibly used as a surrogate endpoint. But the confirmatory evidence piece of this is
Different.
more new to the agency. It's not new, it's been in the regs for a while, but we've put more emphasis on it lately because we understand that in a rare disease space, it's really unusual where you're going to have a single study alone that's going to be robust enough because you've got a small patient population. You're not going to have a large multi-center trial, and that 2 studies may not be possible. So the confirmatory evidence is a way of using some regulatory flexibility to try to substantiate that primary study. And what often happens, and I'll use the tofersen NFL as an example, is that NFL and tofersen wasn't their initial endpoint, but they knew they were going to measure it. And so it's important to have good quality data and assays very early in the design of your studies because you never know what could end up being used as a confirmatory evidence or even a surrogate endpoint in that situation. So if you have promising biomarkers, do the work early up front to make sure that you have good quality assays because you may find that you need to leverage them later for confirmatory evidence or potentially a surrogate.
That's good. Thank you.
Thank you very much for this fascinating presentation and sharing these case studies. I'm Cécile Rivière, I'm the Managing Director for CPAS office in Europe. And particularly in rare disease, as you know, there is a global context that needs to be taken into account. And when you have this situation with challenges around finding the regulatory flexibility, I wonder if you think that there is still an opportunity to develop tools or DDTs that are globally compatible, or at regional level there are with disease symptoms or standard of care differences that would require to focus rather on having DDTs that are focused on a specific region. Thank you.
Yeah, I guess I would say I'm not I'm not aware of what it would really preclude drug development tools from being employed globally. And so I think that we have a lot of conversations with our colleagues across the pond, as they call it, and beyond the EMA, because we are very acutely aware that these are small populations and you don't have the chance for multiple trials over multiple times. So I think that we are happy to leverage any of the work that is being done, within the FDA. We do have a drug development tool program that hopefully many find helpful, and we share that information with our colleagues.
Okay. Last question, I think, for the group. One of the things that's impressed me as I've joined CPATH and end of last year was FDA's commitment to working with the public. There's these individuals are reserved right now, but they clearly on calls have a lot of passion and a lot of interest in the community. And so I wanted to end this session with a bit of a hope. So what things excite you about working in rare disease and get you out of bed and charged to this? And what do you want to convey to the public about that?
Well, I'll start. I think, why we do this is because we see there are significant unmet needs, and, trying to get a product to a population that is going to really change or improve their life. I mean, obviously, we want to make big changes. We're willing to work with small changes as we work there. We want to make this more efficient. But we also, we are the regulators. We balance that because it's just important that we don't put something out there that really doesn't work and an accelerated approval. If you don't get that confirmatory evidence, we don't know. So it's trying to do the right thing so that we can move this field, so that we can get cures, and we can, really work together.
And I would just add from my perspective, I'm a pediatric gastroenterologist. These were my patients. This is what gets me out of bed in the morning. I'm an evolutionary biologist. I believe in moving things forward, always. And so I think the rare disease space for me is just a wonderful way to combine those things, do something that matters. It's a space that is ripe and is necessary for innovation. And I get very excited every day to go to work and try and change things for those that really need it.
Yeah, I can only echo those comments. And, obviously the patients come first, and most of these rare diseases are, devastating diseases that, really impact patients, their families. So striving to find treatments for those patients are always at the first, forethought. But and aside from that, I find the rare disease space to just be challenging, but in a very fascinating way. I love the ability to try to innovate, to try to improve on existing processes, and to try to make trial designs more efficient, to utilize all of the data that's available to us. I just find it endlessly interesting.
I don't really have anything to add, except we look forward to continuing to work with patients and developers and other stakeholders to advance these therapies.
Well, if there's any other questions, we'll close out the session and thank everyone for speaking today. And we'll